Thank you, Doug. So when we started out to plan for this boot camp and also another event where we had a similar topic, we somehow had the goal of providing some kind of guidance to you. Not really a cookbook recipe, but some kind of guidance. Eventually, it was, we really tried, but it's not possible. It's just too complex, and there are too many different compounds out there, and it's just biology. And this is really difficult to predict and press into guidelines. So I think at the end, Doug summarized it well. It still comes up to making a really educated guess, and there are not one or two studies that you just do and then you know if your drug is safe in this respect. So I feel sorry, but there is no clear guidance possible. Maybe in the future we can get together all these experts who are dealing with this topic and agree on a set of studies that would enable us to make a better educated guess. But at the moment, we can just advise of doing as many studies as possible and collect the information and then really come to an educated guess. So some of the questions are dealing exactly with that. They somehow ask us and say, just tell us what to do. I really would like to do that. It would be nice. But it would just not be really realistic to do that. And also for, is there a gold standard for doing certain of these experiments? I mean, we have a lot of literature and we have experience, but there is no document that tells you exactly what to do and how many strains and all these details that we have heard about today. So again, I'm sorry, but probably I cannot answer this question. Another one was, how are regulators dealing with this in vitro data that we are generating? I guess they know as much as we do. That means not enough to handle one or two studies that have been submitted without more information. So it would really be great if you would have a threshold for instance, mutation rate that would tell us and the regulators how to deal with that, but that's also not possible. So one question, and I'm really aware of, especially in early development, that time and money is usually very little. So is there, again, a recipe, one or two studies on the top list? But again, this would be difficult. At the end, I think Doug gave you some indication what would be the characteristics or result of studies that would really ask you to be cautious. But there is no, again, there's no guideline that would tell you when you should terminate a program. So the clinical translation is still needs a lot of work. We have great detailed studies of resistance, but the translation is still in its infancy. And just remember, I mean, the whole resistance topic is not that old in antibiotics. We didn't really care a lot about the resistance in the past. So these translational aspects are still not really well studied. So this was just my comment to some of the questions that asked us to provide guidance on these topics. And we really tried, but I think it would not be realistic to do that. So we have a lot of questions. Some may have been answered already by speakers, but we'll still try to cover as many as possible. And I've organized already the questions and passed on to speakers, but you're welcome all to comment on questions. So I'm asking Mariana to just start with questions that were addressed to her.
Meanwhile, Ursula, there is a big pile there of more questions. So someone asked, mutation frequencies are an important factor. Would you comment in the jump in mutant MICs? And I think both Michael and Doug talked about that a little bit, how you can overcome those mutants, sometimes with dosing, sometimes with trying to find more potent like derivatives of your compound. The other question was, since we don't like the term spontaneous, let's change it. I agree, like, let's change it. I don't know how to start. We can talk about that. Do epidemiological studies teach us everything about the risk of resistance? I think running a surveillance program for so many years, I think so. Like, I think when you look at your population and you see already some resistance in the isolates before you even launch your drug, it's concerning, and I think a lot of people prefer to not take the risk. However, it takes some time for when the drugs start being used and we see resistance emerging, it takes some time for us to pick that up in surveillance, just because we have to collect a limited number of isolates for different hospitals. But that's the reason why both the FDA and the EMA now consider the post-approval surveillance of up to 5 years, and sometimes even more. The EMA states that they can prolong that to 10 years of surveillance, post-approval surveillance of clinical isolates to evaluate emergence of resistance. So surveillance is an important tool for that. Another question that was related, how do you have an assay to screen mobile element, like resistance mediated by mobile elements? And that's really hard, because you can postulate a few things. You can look in the wild type, like the isolates before your drug is released, to evaluate that. But it's hard to predict what's going to happen in the clinic due to genetic elements. It's a very complex task. So there is someone that asked for all of us, what do you think is the best way to evaluate biological fitness, if you do that in vivo or in vitro? And another person sent a more, another question saying, since this studies don't mean anything or don't translate, why do we keep doing them? So I agree with the other two speakers. Like, you were trying to collect as much information possible to put a picture together of what you think might happen or you think are the signs for you to not proceed. But it's impossible to predict exactly what's going to happen. I don't know if you guys want to comment.
Well, I mean, so this will be interesting to see where this goes. But in terms of assessing fitness of mutants, I mean, there's very simple ways to do it, that you can do growth rates, or if you want to get a finer measure, you can do competitions where you mix a wild type strain and a mutant strain, and you assess the ratios of them as they passage over time. And then you come to the question of, well, what's the relevance of doing this in the laboratory? And the answer is sometimes it seems to translate quite well to the clinical experience, that some low fitness mutants in a test tube are low fitness inside an animal or a human as well, and so that it's very good. But then in some cases, like the Mesolinum example, it gets more complicated, where the fitness doesn't quite encapsulate everything in Muller-Hinton broth because they're growing in urine in a particular location, and maybe it's different. And so it's not going to be able to answer anything. And so then if you want to get a more complex answer, then I think you have to go on to what Michael was talking about, which is assessing them in an animal infection model in various forms. And I maybe you want to...
Oh yeah, I think as you said, I mean, you can the in vivo fitness can be performed usually in the animal model. In mice is probably preferred, and as the competitive index is usually, because sometimes you evaluate the in vivo fitness by doing an LD50, which is not the right way of doing it. Here you really need to have a mixture of 1-to-1 wild type versus mutant, and then over time see if your mutant is really less fit and disappears. I would say also to try to do that under selection, under treatment, makes a lot of sense, because then you can directly assess whether your mutant will indeed be so badly damaged compared to the wild type that it will not be able to overcome under pressure your wild type. But then, does that translate in the clinic? Nobody knows. Will it be the same whether you do a urinary tract infection, a pulmonary infection, or a septicemia? Nobody knows.
Do you think that we could use the in vitro to screen and then do a more stepwise approach, like you do the isolates in vitro tests if they have different fitness, and then go to an in vivo model that you would reduce the cost a little bit? So maybe a stepwise approach would be, it's how you could use the in vivo versus in vitro?
Yeah, definitely, and I think also it maybe an answer to why we do these experiments, especially the simple ones like frequency of resistance, if they don't mean anything. I think when you ask the question, you're a bit biased because you're thinking of one compound, but usually when you have a medchem program, you have several compounds. And so if you test, 3, 4, 5 different compounds, why would you pick the one that has a lousy frequency of resistance when you have the next one that are better frequency of resistance? That doesn't make any sense. So I think this is a bit why we do that. It's because we can do them, because we can make them, some of them can be made very easily, and because we have to choose between different compounds, and this is, an argument to choose. But then, yeah, and also since there are very different levels of complexity, you can do them in stepwise manner.
I would like to come back to the Mesolinum example. Should the in vitro experiments be done in urine or blood, depending on the planned indication? Would that make sense in general?
I mean, yes, there's a recent study that came out that did exactly that and said, perhaps we should be doing all of these assays in urine. But the fact is, at this point, everything is quite standardized that, everyone does MIC assays in largely similar conditions. And so, yeah, maybe that would be an adjustment that would make sense to make in specific cases. I mean, it sort of opens up a broader question. How useful is a standard MIC for every compound and every TPP? And I mean, it could get very big very fast. But
not only that, how are you going to compare? Like, I agree. You probably should proceed with doing that experiment in a body fluid. But how do you compare if you don't do it with a standard methodology that everybody uses? How are you going to compare that and have an idea of what's good and what's bad? So I think that in the end, it's another step and not let's replace everything for urine or serum or whatever.
Well, and even saying urine, it turns out that the type of urine matters quite a bit.
Exactly.
That some urine samples, even pooled urine samples, you see different effects. So trying to imagine standardizing that seems a little, yeah.
There was just a more a comment than a question about thresholds of regulatory authorities, and a big no. And I think that's not even planned because we don't know enough, we don't have enough information. So there are no thresholds issued by regulatory agencies. So that's not a concern.
Someone asked also if these lessons apply to antifungal drugs. And I have been working with antifungals for several years. Yes, but there is the limitations, like antifungal yeasts and molds are eukaryotic, and most of them have multiple copies of each gene. So, and they don't acquire resistance. So you have to think about the differences of that as well.
I have two quick one. One that I said at one point I said you need a lot of drugs to do the spontaneous frequency of resistance, and then someone asked how much. So if you're, of course, the worse the MIC, the more compound you need. But I would say for a compound that has an MIC about 1 microgram per ml, if you want to do it well, you need about 5 to 10 milligram of the compound. So that's not a lot. But usually in a medchem program we get the compound in a couple of milligrams to do the MICs, and so you cannot do the MICs and the frequency of resistance study. And of course that's for 1 microgram per ml. If your compound has 10 microgram per ml, then it's 50 to 100 milligrams. And then I think one that I really like is like, if resistance is not found, does that mean it will never be found? Or what can one infer from data where resistance is not observed? That's a good one. To be honest, if you have very low resistance, if resistance is not found, I think it says something about your target. Maybe that you don't have one. I think it's a case where too much of a good thing is really bad. So if you really don't see resistance, no, seriously, it could be just, that we have the example of teixobactin, for instance. It could be that your target is really something that is very hard to mutate and would require many, many genes and so forth. But usually, yeah, I would say when we see something that has absolutely no resistance, it usually means not specific.
Michael, there was a group that developed an assay trying to understand better the mutation rates on linezolid, and they developed an assay specific for drugs that have very low mutation rates, and so that's an option too. It's a kind of, it looks a little bit complex assay, but you could go to a more target type of assay. That's not what we perform more commonly.
So there's one question related, and that's the example of penicillin and A streptococci. So is it merely lucky, or can we learn from this biology to identify cases where resistance will be very rare? I think there were some theories related to the size of the genome or so, but in general, I'm not sure if we know and if we understand why this specific case was so successful in the history of antibiotics.
Going back to the linezolid history, that is an interesting one just because I think we talk a lot about that at where I work. You have 7 copies of the ribosomal RNA gene, and you have to mutate multiple copies to have resistance. So I think also that could count into why it's difficult to generate resistance and how to translate that.
So Ursula has handed me a question here. Lots of reference to fitness cost. Is there an easy way in vitro to measure this? And yeah, I think the easiest way is to just measure growth rates. So you can do it in a flask, you can do it in tubes, you can do it in a fancy machine, but ultimately that is a way. You can't get extremely accurate numbers, but it's going to give you an idea. You're going to know, do you have 10%, 20%, 50% in vitro fitness cost? And then from there, you can make further decisions about follow-up studies. if these are very unfit, maybe you don't have to do a lot of follow-up studies because you say these are very sick strains and probably aren't going anywhere. But if they're 90% fit, then you have a difficult situation. I will just do another one here. Using Mesolinum as an example, can you comment on how this compound would, quote, survive through resistance evaluation in a part of an active medchem program? And I think that that's a really good question, because I look at Mesolinum and I'm not sure that it would immediately jump out to me that this would be worth pursuing based on the frequency of resistance just because it's so high. And so I don't know, does anyone else have thoughts on what they would do to convince themselves that they should pursue Mesolinum?
I mean, there are more drugs in development that are PPP2 inhibitors, and they are all developed in combination because they showed exactly the same problem in development. So I'm not sure if it would be such a good idea to just try and develop them as a single agent, especially because they are not only targeted at urinary tract infections.
And maybe that's the key, that Mesolinum has a particularly well-suited TPP for it, but still the same question, how would you have known?
How would you know? Yeah, how would you know? It's complicated.
And we also don't really know what would have happened if it would have been used really widely. So selection pressure was quite low.
I think, like related on your comment on UTI, I have a question. Should frequency of resistance benchmark be different for different indications for use? For example, would you have some benchmark threshold in frequency of resistance for UTI versus HAP-VAP? Again, very, very good question, and I have absolutely no idea. I think usually in your development for a typical gram-negative compound, you have to go through UTI, and so in which case, it's an example where you might encounter a very high inoculum size, quote unquote, and so I would say bear for the worst, but I'm not sure.
Just to follow up here, I mean, I wonder, keeping with the theme, I have one here that I'm not sure that I can address, which is, should clinical dose be based on multiples of MIC that give below a certain frequency of resistance only? And I mean, what I've seen is practically, oftentimes the dose has almost nothing to do with the frequency of resistance. It's not considered. What you're looking at is how tolerable your compound is and what the effective dose is. Now, I suppose you could find yourself in a situation where the mutant preventing concentration is very close to your dose, and so you decide to go higher. But it seems like there's so many calculations that go into that frequency of resistance tends to be pretty low on the priority list, is what I've seen.
Yeah, so here I've got quite a few questions around that. Should we consider MPC, mutant prevention concentration, while doing PK/PD, time above MPC, AUC divided by MPC, etc.? Can you discuss the value of the MPC in evaluating mutational resistance? Any clinical correlates of these concepts? Can you comment on the determination of MPC, etc., etc.? So I think this is a recurring theme, and how does dosing and the MPC concept chime into your development program? And here, I've seen in a few programs that dosing regimen has actually an impact on mutant selection, and I think I would refer to a number of PK/PD reviews. The one by George Druzano, Keeping Our Powder Dry, I think is great to show examples on how changing the dosing regimen and mimicking using hollow fiber PK/PD, you clearly see that you have an impact on how easy it is or hard it is to select for resistance and have this resistant mutant take over. So clearly these concepts I think are important, and so if your MPC, for instance, is really achievable, I think this is an instance where you have to shift your programs with that in mind, so meaning that you will have to dose higher.
One comment that I got when I was talking to different people about what concentration should we use to do the frequency of resistance, if we should use your peak, your trough. One thing that people told me was that early in the development, when they usually do the mutation studies, they don't have that information. And do you think that we should maybe perform the more traditional experiments early in the development, and then when you have something similar to your dose, like that you have an idea of your dose, then maybe retest some of those strains in a PK/PD setting?
Yeah, I would turn the question around. So meaning, for instance, if you have an example where your compound has a frequency of resistance that you think is bad, the program has that has a bad frequency of resistance, then you could suggest immediately to do to go how high can you go in vivo? You know, what is the PK in vivo that you can achieve? What is the highest that you can achieve? And then repeat the frequency of resistance experiment using those numbers, having those numbers in mind. And see if there is a difference of frequency of resistance in the range of concentration that you can achieve by dosing, like say 3 times, 4 times, and so forth. And if you can, then when you do the experiments and you see that, oh, in those cases the frequency of resistance is not that bad, then that's a path forward, I think.
Yeah, I have a more general question. Isn't it reasonable to assume that a single target will give resistance and then do experiments to show that they don't? So assuming from the beginning the single target drugs will have a problem, that would be then the next question. Should it be incorporated into a drug discovery project to always cover 2 binding sites, 2 targets? Would that be more on the safe side? Has someone a comment?
So meaning killing every project that is a target-based project?
Just one target, yeah. I don't think it's realistic.
I mean, maybe coming at this from a different side here, actually, I have a couple of questions here about doing multi-drug treatment, so treating with multiple drugs at the same time. And there's some questions about the regulatory elements of that, and I can't really address that. But I will say that one of the things that makes a lot of the organisms that we work on different than, say, antivirals or even MTB is the complicating factor of horizontally transferred resistance, that treating with multiple compounds can sometimes just force the assembly of really nasty plasmids. But again, the regulatory questions I can't really address.
We have some questions related to polymicrobial infections. How should that be addressed?
I don't think we have a good answer for that. Polymicrobial infections are already so complex, and how are you going to mix that up into an in vivo study and answer those questions?
Yeah, probably just testing them individually and then draw some conclusions.
Yeah, I think it's there's no way, to my knowledge, to address directly the polymicrobial infection. Only multiplication of doing the studies in the various species and bearing that in mind. I have something that is related to polymicrobial infection, but is are you controlling the microbiome in your experiments? You know, these days everything has to be looked at from the microbiome perspective. But so, what microbiome-related effects do you see? Does diet changes the susceptibility to resistance? All very good questions, but frankly, I have no answer to that. You know, so.
One question is, if you know of any example that a developmental agent that had low mutation frequency and then still failed clinically.
Due to high development.
Yeah, due to resistance emergence.
I'm not aware, but.
No. So we don't know any example. If someone is here who knows, then please get up. Do we have other questions? questions?
I had a quick one, which is why is, related to my talk, why is influx harder to overcome than efflux? I think this is because usually we target, when we talk, we target gram-negative bacteria, and in that case, the rules of penetration are still not understood. So there are a number of things that have been proposed, but they are not so clear. And so, for instance, you might want to add a primary amine to your compound. This usually works to improve the influx, but it's not. There is no consensus. Rigidity, globularity of the molecule are also parameters that have been suggested to be important to augment the influx, but I think there is no consensus. So that's why. It's because the rules of penetrations are not understood for gram-negative bacteria.
I have a quick one here as well. How to assess for acquired resistance? What about assessing the level of already existing mechanisms? And so that's fairly straightforward. It's MIC90 assays. So you get a large collection of current clinical isolates and you just directly test them. And that's going to give you a good idea whether there's some big resistance mechanism floating around out there.
And so I have an example here where one of the resistance mechanisms was actually in a commensal bacterium. So, which is not usually something you test for, but then in that case, and for instance, you really wonder, what's going to happen when you actually treat a patient. And there is also examples of compounds, antibiotics compounds that are actually coming from the commensal bacteria. In that case, you really wonder what is going to be their horizontal gene transfer contribution to resistance for pathogens.
So out of curiosity, was that just you were screening commensal organisms to just keep an eye on them?
No, it was just, it's something like Listeria innocua versus Listeria monocytogenes, and so it's one of those instances.
So Doug mentioned the factors that would raise your red lamps, and there's a question. Should CARB-X and other funders set a policy not to fund a drug in development that has these three red flags: high frequency of resistance, high fitness, and high shift in mutant MICs? Should there be a policy?
I mean, I suspect, whether it's explicit or not, I suspect that it's already there. I mean, this is something that you would consider with a compound. Any class of compounds that you're looking at, these would be some of the things that you would look at. And if you got all three of those, I think that would be a suggestion that maybe it's not worth pursuing that compound. But of course, there's lots of different things that go into it. Maybe it's extremely potent, so I don't know.
Yeah, and most of these programs would not have this information when they submit for funders, for funding.
I've got one that is looking at the mode of resistance of an antibiotic producer. Is it a way to predict the mode of resistance of the pathogenic species or strains? That's a good one. Sometimes it does, like vancomycin. Sometimes it has absolutely no bearing. So it really depends. No quick answer to that one.
So should you use clinical or laboratory strains to measure frequency of resistance? And I find lab strains are great because everybody has access to them, but sometimes they have been in the lab for so long that they are not representative of what's in the clinics now. So I think a mixture of them is a good model, and I don't know about what the others.
Yeah, I agree there, and the frequency of resistance of the recent clinical isolates sometimes are very discouraging. I was very surprised. Like something that is highly resistant, highly virulent, and you have absolutely terrible frequency of resistance compared to a lab strain. You need that information.
I mean, I would just add that whenever we're working on programs, generally it's nice to start with something that's well behaved. And so a lot of times I'll start off with a laboratory strain just because then when it comes time to do whole genome sequencing, I have a good idea that I'm going to be able to interpret the results, as opposed to starting with something that's not as well characterized. The identifying the resistance mechanism can be quite a lot more difficult. And that's different than some of these things that you're talking about that are later on down the road, where you're talking about, well, what is the frequency of resistance in multiple organisms? Then you're talking about a lot more work, and hopefully you've already done it on a laboratory strain, so you have an idea of what you might be able to expect.
But every time that we try to design these experiments, you choose like a couple of strains because, as Michael mentioned and you mentioned in your lecture, no strain behave exactly like another. So you tend to choose like 6 to 8 isolates to do your mutation frequencies. And in that population, probably you can include a few control lab strains and a few clinical isolates. That I think we should keep in mind to try to answer the questions that are important to your drug. Like, is your drug geared towards beta-lactamase producing strains? So try to do with isolates that are representative of that, not like a super susceptible E. coli that might never be treated with your drug.
I have here 2 questions that we may not be able to answer. One is a drug discovery strategy question. How would you search for a dual target agent compound? I guess that's a drug discovery strategy, and some companies really claim that that's their goal to do, but I don't think we can address this. Another one is we may also not be able to address. There is more out there now in development than traditional antibiotics. And we have one question related to bacteriophages, how to characterize that. And I think that's a very specific question and a very relevant one, because resistance is a major issue when there are bacteriophages in development, and that's also the reason, one of the main reasons for applying cocktails.
Yeah, even though I would refer you to read the results of the Fago-Burn clinical trial. It was interesting because in that case, the people who are treating burn wound infection by Pseudomonas aeruginosa with a cocktail of 12 phages, and in 40% of the cases, there was resistance that occurred to the whole 12 phages. That was very discouraging also. Probably a dose effect. But.
Yeah, I think this study had some issues, but resistance is a main one.
And for virulence, just to some compounds that are directed at virulence markers or capsule or wherever you can have resistant pressure, you can oftentimes select for resistance mutant. For instance, a mAb that is opsonophagocytic. You know, people will say that it's harder to develop resistance. Well, you should try it, and you would be surprised. I mean, I have a couple of examples in mind where it's very easy to actually see resistance appear, and then you would think, oh, but surely the mutant is not going to be resistant in fit in vivo, and sometimes you have very odd results.
Yeah, so that's when I realized that also when I researched the literature. So usually antivirulence compounds are considered as not prone to resistance, and it's not an issue at all. But then you find the studies that show exactly the opposite. So you are not on the safe side if you are going into the antivirulence sector. Do we have any?
Was not answered. So what's the best way to evaluate resistance that arise from changing protein versus genetic mutations? So usually protein expression is upregulated or downregulated by promoters, by other genes that can have regulators or promoters that have mutations. So if you do enough sequencing and understand the pathways, you can look for those mutations, or you can look at expression assays, or more recently people are doing whole transcriptome that you can look at expression of several proteins at once. And you can compare with the parental strain. And that's a question for both of you that I had during your talks. Like, you don't recommend using whole genome sequencing, but you could compare with your parental strain And we actually have done that in the past. Like you see, you de novo assembly your parental staining and your mutants and compare it, SNP, and do a SNP analysis. So I think it is a method that can be used.
No, actually, yeah. So I said you should absolutely do the whole genome sequence when you do what you just mentioned. So starting from a parent strain, you select for resistant mutant, do the whole genome. I mean, it's fast, it's easy.
And nowadays cheaper than it was before, yes.
And it gives you a lot of information. So no, you should absolutely do the whole genome. Doing the whole genome on a clinical strain, when you have, and you do the MIC90s and you've got like 4 strains that have high MIC, getting the whole genome of that, usually not worth it. Because you have, unless you already know what to look for. Because
I sequence a lot of clinical isolate, so.
No. Then you're getting into territory where you have to do genome-wide association studies and have to sequence. You have to sequence sensitive strain, resistant strains, also, and compare the whole gene or many genomes, and then you will need a lot of strains to do that. I've got a couple of questions that I've related to the in vivo models. What about measures of frequency of resistance in vivo in animal models? And could an in vivo model have been able to distinguish the GSK compound and the meselinam and predict the clinical emergence of the resistance to the GSK compound? So frequency of resistance in vivo, I think the whole concept, I mean, just the sentence is wrong. It's not a frequency of resistance the way we do the spontaneous frequency of resistance, because there is the PK that makes the concentration of the compound fluctuate, the bacteria are growing, depending on, there are so many parameters that you cannot call that even a frequency of resistance. So, I've shown you an example when we do the double plating, when we see that the mutants are actually appearing in vivo. That's so the way we do it, I think, is not normalized, and so I don't call that a frequency of resistance. I think there are very few, I've seen very few examples of people actually doing this kind of experiment. I've seen some people who are doing cage experiment to have an in vivo situation and a control inoculum to have more access to on the parameters. But I think it's very early days and we are very far from a consensus. But I would really encourage you to do the experiment because it doesn't take much. When you do the efficacy studies, do the double plating, and it might tell you something. So related to the question about would in vivo, if we had done the in vivo model, pushed the in vivo model, would have we seen anything with the GSK compound? I think in retrospect, when the GSK compound was assessed using hollow fiber model, just looking at the regimen of administration showed that you could have seen that the GSK compound would have quickly selected for resistance. So I think if it had been done in vivo, maybe we would have seen something, but I'm not aware that this has been done.
Yeah, I think we had a lot of interesting questions, and it just shows that we still have a lot of gaps with translating in vitro information and basic research information to the clinical situation. And maybe it would be a really good opportunity to ask for specialists coming together and discuss more details and give more guidance and develop some standards for basic assays. Yeah, with this, I would like to say thank you to all of you and all these interesting questions. We keep them in mind, and maybe there's another opportunity to have more discussions. Thank you very much for your interest.